So I'll tell you today about our work on neural codes for natural behaviors in flying bats. So as David indicated, I'm also interested in the neural basis of behavior, in particular natural behaviors. But I often ask myself, which behaviors should we study? So for example, if you're interested in social behaviors or group interactions, then for example, this kind of behavior, group hunting in lions and communal defense in buffalo, is very interesting to study. There's a lot of things going on here. But you might say, well, it's not so controlled, it's hard to study. Also, there are obvious safety issues here. So most people interested in social neuroscience opt for studying something like this, tube test in mice. And so we have here this perception of, on one hand, controlled laboratory experiments, like on the left, that are controlled but perhaps unnatural. And on the other hand, we have natural behaviors that are natural but uncontrolled. And I would like to argue today that this is this sort of dichotomy, or this separation is not so simple, because for one, the behavior by the lions and the buffalo is not so uncontrolled. They're not just running randomly around. They're repeating certain hunting behaviors over and over. So there is some sort of internal control that the animal is doing by repeating the behavior. And on the other hand, this behavior is not totally unnatural, because mice are burrowing animals. So to go in this kind of burrow-like tube and encounter another animal is not totally unnatural. So the picture is more complicated, and I'd like to argue that there's actually kind of a two-dimensional axis, controlled and uncontrolled, and artificial versus natural. And so, of course, you can have extreme situations, like an experiment on head fixed self-stimulation, which is very controlled but highly artificial. And on the other extreme, you can have 24/7 unconstrained behavior, very natural but totally uncontrolled. And sort of this line that connects the two is this axis that I showed in the previous slide. But then you can have things like this, birdsong, for example. So zebra finches in particular repeat the same song over and over hundreds of times a day, and with the precision of a few milliseconds. So on one hand, it's a highly natural behavior. On the other, it's also highly controlled. The animal is doing the control for us by repeating the exact same thing over and over again. And the lion hunting is maybe here. It's not as reproducible, not as controlled as birdsong, but it's also in the upper right quadrant. So I would like to argue that this is sort of the area of this phase plane that we want to reach when studying the neural basis of natural behavior, or behavior in general. And so in my own research, we studied the neural basis of navigation, and we're also trying to get there. And basically today's talk will be to share with you some of our journey trying to get to this sweet spot of both natural and controlled behaviors. So a few words about the neural base of navigation, about what we know about the neurons involved, and I'm sure most of you know this. So in the hippocampus, originally of rodents, but now of multiple species, there are place cells that were discovered by John O'Keefe. And in this example, if you let a rat run in a 1 by 1 meter box or so the top right shows the trajectories of the rat in the arena, and the red dots are spikes. And below is sort of a heat map, or showing the place field of this neuron. It likes to fire in a particular location in the environment. So O'Keefe called these place cells because they seem to represent the position of the animal. Different neurons fire in different locations in the environment. Then in the medial neuronal cortex, there are grid cells discovered by the Mosers, neurons that not fire in one position, but in multiple positions, creating a kind of hexagonal tessellating grid. And then there are also head-direction cells, neurons that don't care so much about the position of the animal in space, but about the direction its head is pointing to. So we have sort of neural maps or cognitive maps, neural distance meters in the grid cells, and neural compasses in the head-direction cells. And together, they underlie, we think, the neural base of navigation. Now, what we ask in my lab is, how does real-life navigation differ from navigating in these small laboratory setups? Because typical experiments are done in these 1 meter or so boxes, or on short 1 or 2 meter linear tracks. So how does real world navigation differ from that? And so you can list several answers. One is that the world is not flat, it is complex and three-dimensional. The world is not 1 meter in size. Navigation occurs over kilometers. How does that affect the system? There are also rapid switches between navigation and other behaviors. So how does that modulate the network? And also navigation often occurs in groups. How do these social factors come into play? You can give more answers, but these 4 answers are things that we have been investigating actively in my lab, and these will also be the things that I'll be telling you about today. And now we studied this not in rodents, but in bats, Egyptian fruit bats in particular, and I think it will become apparent as we go along why bats. The idea is, of course, not to understand the bat, but to understand the brain. But as I'll show you, you can use bats and their interesting behaviors to ask questions that are difficult or sometimes impossible to ask in animal in other animal models or in humans. So they have interesting advantages because of their behaviors. So Egyptian fruit bats are big bats, so they're much bigger than what most people are used from Europe, for example. 150 to 180 grams. It means they can carry a lot of weight in terms of neural recording systems, etc., as I'll show you. They also, by the way, have great vision. This particular species, you see they have big eyes. So blind as a bat is a misnomer, but certainly for this species, it's not true. They see very well, and they also have a sense of echolocation or by a sonar that I'll show you a little bit later today. So just to put you in the mindset of what does it mean to navigate for a bat, here's an example from a study that we did a decade ago, where we put little GPS data loggers on the back of a bat just to track the navigational trajectories of wild bats outdoors, just the behavior. And we found basically that the bats that are cave-dwelling bats fly from their cave to a particular fruit tree, a preferred fruit tree, night after night for many nights. So in this particular case, all of this is data from one animal. Each colored line is a different night. And you see that this bat really flew in a nice flyway from the cave to the tree night after night. Of course, after a few weeks, it might switch to another tree, but for a while, at least a week or 2 or 3, it will fly in this very nice flyway in distances of 10 or 15 kilometers typically, and sometimes more. So these are the kinds of behaviors that we'd like to eventually understand, and these are the kinds of spatial scales we'd like to eventually understand. But I'll start from what happens in 3 dimensions. So 3D spatial codes in the bat brain, 3D place cells, 3D directional cells, and 3D grid cells, work by PhD students, past and present, Michael Yartsev, Arseny Finkelstein, and Gili Ginosar. So before I tell you about what happens in 3 dimensions, in 3D, I'll just tell you that in 2D, when we sort of ratified the bat, let the bat crawl on 2-dimensional surfaces, we found the same types of neurons. So we found place cells, grid cells, and head direction cells in 2 dimensions, similar to what you find in rodents. And when we went to 3D using wireless neural recording devices, Michael Yartsev, when he was a PhD student in the lab, found 3D place cells. So we have in the lab about a 5 by 6 by 3 meter flight room that you see here. And this particular neuron was strongly activated whenever, this is neuron in hippocampus, it was activated whenever the bat flew through a particular 3-dimensional volume. This is the red area, a particular three-dimensional volume about 80 or 90 centimeter in size, and this area was spherical, meaning the resolution of the representation is the same in the X, Y, and the Z dimension. A couple of years later, Arseny Finkelstein asked, are there 3D head direction cells? And the answer is yes, there are neurons that represent the direction of the head in 3 dimensions, so whether the bat is looking at that direction or that direction or that direction, etc. Interestingly, he was able to show in that paper that these head direction cells do not represent 3-dimensional directions in the classical textbook spherical coordinate system, but in a toroidal coordinate system. And this gives interesting, actually biological advantages in terms of computation. I won't go into this today, but you're welcome to check out this paper of Arseny. And just to say that recently several other groups found very similar neurons also in other species. So 3D head direction cells were found in monkeys and in mice, 3D place cells in rats. So I mean, certainly monkeys are very much 3-dimensional animals, moving between their branches, but also mice are great climbers. So it's nice that that these types of representations are not just in bats, but seem to occur across mammalian species. What about 3D grid cells? So here, Gili Ginosar tackled this question. And so in 2 dimensions, this is an example of a beautiful 2-dimensional grid cell from the Moser lab. Grid cells are characterized by 2 properties, 2 main properties. There are several more, but I'll focus on these 2 properties. One is their famous hexagonality, this hexagonal lattice that tiles the entire environment. And secondly, they have a fixed distance scale, meaning the activity fields or the activity regions that are nearby each other all have kind of fixed distances between each other. Now, you would immediately say, well, it's sort of the same thing, right? If you have an hexagonal lattice, you'll also have fixed distances. It's sort of geometry, basic geometry. And it's true for generally for 2 dimensions, but as I'll show you in 3 dimensions, these 2 properties, the hexagonality and the fixed distance scale, become separated, and it's important to look at each of them separately. So what might we expect for 3D grid cells? On the one hand, we might expect to see global lattices, like seen here. So either maybe cylinders that are creating a 2-dimensional lattice, but they're sort of elongated vertically, or maybe a hexagonal type of a lattice, like here, of spherical activity fields, or maybe some other lattice, like a cubic lattice. So various lattices on one hand, and on the other hand, could be disorder of fields, and could be something in between that is not something in between order and disorder. So these possibilities in mind, Gili, to do the experiments, so she recorded the medial neuronal cortex of flying bats in the same 6 by 5 by 3 meter room, flight room that we have, and she put around the periphery, around the perimeter of the room, really close to the walls, between 6 and 11 landing balls that had different heights. Some were low, some were high, and this basically encouraged the animal to fly at different heights and to span really nicely the 3-dimensional volume of the room, which they did. And she used a wireless electrophysiology system to record neurons from medial neuronal cortex. And so they flew quite extensively, covered the room. The bats covered the room quite nicely. This is data from one session. On average, they flew almost 2 kilometers per recording session. And so here are examples of 8 neurons that Gili recorded from the neuronal cortex in these conditions. And so you immediately see that you see 2 things. One is that these neurons have kind of activity regions or activity fields, grid fields, and that these fields seem spherical, sort of like the place cells that I showed you, where there was an individual place field, but here there are multiple of them. They don't seem to be elongated. So you can already, just by looking at that, you can rule out this possibility. We did it more formally. We quantified the elongation, and indeed there wasn't any systematic elongation. So the fields seem to be rather spherical. The other thing is that you can see from here that it's really hard to see is there a lattice here or isn't there? So we need to quantify things. So we detected kind of stable and reliable fields. I won't go into the detail, but we don't want to record any to detect any little local peaks. So we add some conditions on what is a stable and reliable field. I won't go into the details. You can have a look at the paper. It was published just a few weeks ago. And overall, we had 66 cells with more than 10 stable fields. And so we focused on these neurons with at least 10 stable fields as candidates for grid cells. So first thing that we did is to look at these local distance property. Are the activity fields of neurons, the nearby activity fields, do they have a sort of fixed distance scale? And so what we did is we looked at an individual field and then found its 3 nearest neighbors and collected these 3 distances, then went to the next field, measured its 3 nearest neighbor distances, of course, not counting distances twice. And then we can construct a histogram of these local distances, nearest neighbor distances. So this is an example for this particular neuron. You see the histogram of the interfield distances. And what you can see from here is this histogram is narrow, meaning activity fields tend to have fixed local distances from each other. And you can quantify how narrow is the histogram by using the coefficient of variation, standard deviation, divided by the mean of this histogram. And for this particular neuron, the coefficient of variation is 0.17, which is a small number. It means it's narrow. And importantly, it's smaller than the coefficient of variations for 1,000 shuffles. So from that, we conclude that this neuron really has this local distance properties. And indeed, 20 to 30% of the multifield neurons had this local distance scale. So they were 3D grid cells. Next, we asked, do these neurons show a global hexagonal lattice like seen in 2D? So can we find something like these optimal space packing lattices like FCC or HCP lattices? For those of you who know about this, these are the optimal way to pack space with spheres, as shown here. And the very short answer to a very long journey is no. So we did a lot of different things. I won't go into the detail because we don't have time for this. But basically, we were able to show that there are no lattices. So just to give you a flavor of what we did, one of the analyses that we did is to fit various of these FCC and HCP perfect lattices. So to run through millions of possibilities. So the brute force find the best fitting lattice to each neuron, and then compare it to his 1,000 shuffles. And you see that for this particular neuron, the actual fit error for the FCC was not different from the shuffles. And this was true, in fact, for all the neurons. So we didn't find really any neurons that were convincingly and significantly had a global hexagonal lattice. We also looked for other passable lattices by looking at distribution of local angles, but this distribution did not differ from shuffles. So again, arguing against any other lattices as well. So we can conclude from that from what I just showed you, that we don't have this type of arrangement because we don't have these elongated fields. We don't have a hexagonal lattice. We also don't have any cubic lattice or any other lattice because of the distribution of angles. On the other hand, this is not a total disorder because we do see these fixed local distances. So it looks like we have something in between order and disorder. We have this local order, these local distances preserved. And so next, to really understand what's going on and why is this difference between these beautiful hexagonal grids in 2 dimensions and this local order that we find in 3 dimensions, we teamed up with a team of theoreticians, Yonatan Al-Khadef, Yoram Burak, and Chaim Sompolinsky, who did some modeling work. So basically, their approach to this was to treat the firing fields of grid cells as particles. And there's a long history in physics and chemistry of looking at complex systems with particles that have interactions between them. And then depending on the properties of this interaction, you can get anything from complete order, like on the left, to complete disorder on the right and things in between. So basically, we wanted to see whether this interaction between fields can explain what's going on in 2D versus 3D. So we used a particular interaction potential called the Lennard-Jones potential. It's widely used in physics and chemistry. This is the form of it. So this is the potential versus the distance between nearby fields. And it's this interaction potential is characterized by repulsion at short distances, meaning fields that are too close repel each other. Fields that are more far away attract each other. And in between, there is a kind of optimal distance. But then there's also noise. We had noise or a temperature that introduces fluctuations in field positions and move them away from a perfect arrangement. And then it turned out that this type of a simple model, by the way, just to say that such a type of a potential can arise in a neural network model. So although our model was phenomenological in nature, we basically used a mathematical equation, but this type of or similar type of interactions can arise in neural networks with competitive plasticity. So it's biologically plausible. And what we found is that this type of model explained the data much better than random Poisson. So if we look at the particular distance metric between the data and the model, I won't go into the detail of this distance. We found that the pairwise interaction model, the distance was much smaller, meaning the fit was much better as compared to random Poisson. That's just another way, random Poisson arrangement of fields. That's just another way to say that the fields were not arranged randomly. We also compared to jittered FCC or jittered HCP, sort of considered the possibility that this is biology. We perhaps cannot really expect perfect lattices. So maybe there are these lattices, but they sort of are jittered. So we modeled this possibility, and again, the pairwise interaction model explained data much better than these jittered FCC or jittered HCP models. So we can really conclude that this pairwise interaction model captures the data much better than the alternatives. What happens in 2 dimensions? So why is there a difference between 2 dimensions and 3 dimensions? To try to understand that, we use the radial density function, which is a way to capture distances in all scales. So the intuition is that, imagine this perfect lattice as you see on the left, the honeycomb. So the distance from every activity field to its nearest neighbors, if you look now at distances to all possible cells of the honeycomb, so you have a peak at distance 1. And then further apart, you have a peak at distance 2. So basically, if you have a perfect lattice like the honeycomb, if you look at this distribution of distances to all possible sort of all-to-all possible pairs of particles, you'd get a distribution with multiple peaks, including a distance 2 and 3 and 4, etc. And when we did that for the 3D simulations, then we did not find a peak at distance 2. This really decreased, and there was no peak at distance 2. So really, there's no indication for a lattice in 3D. However, when we did the same for 2 dimensions, so basically carried out simulations in a squished room. So basically, we took the room and squished it to a 10-centimeter height. So it's still 3-dimensional, but it's quasi-2-dimensional. Then, indeed, a peak at distance 2 and 3 and 4 emerged as expected. And in fact, in these 2-dimensional simulations, we found many cases of particle arrangements that looked really like 2-dimensional grid cells. Whereas in 3D, it was much less organized, had this local distance scale. So this really shows that this exact same model really captures both what happens in 2D, the global hexagonal lattice shown in 2D, and the local but not global arrangement that we observed in 3D. And again, to give you an intuition, you might be perplexed. Why is this difference? It turns out in physics that it has been known for many years in what's called jam packing of spheres. So if you think about the following scenario, if you take a lot of spheres and put them on a surface of a table and try to pack them together, we'll always get a kind of hexagonal lattice, as you see here. Anybody who played billiard or pool knows what I'm talking about. On the other hand, if you throw a lot of spheres into a 3-dimensional box, So like a box of marbles or a box of balls in a gymboree or whatever, like something like that, you'll never get a perfect signal lattice. The balls will always get stuck in a local energy minimum. However, if you look at the particular field and in its distances from its nearest neighbors, the distances will be fixed. In this case, they're exactly equal to the diameter of the sphere because they're jammed, they're stuck together, right? So this exactly shows that this type of arrangement is what on the right is what you see in two-dimensional grid cells, and the type of arrangement on the left is what we see in the 3D grid cells, the local distances but without a global metric. So to summarize this story, we found cells with multiple 3D fields. 20 to 30% of them were grid cells having a local distance scales but no global lattice. I'm saying 20 to 30% because 20 versus 30% depending on which type of shuffling we did. We'll go into the details of that. We suggest that a fundamental property of grid cells is having multiple fields with a characteristic pairwise distance between them. So not the hexagonality per se, but actually the fixed distances between fields, that's the fundamental property. And in 2D, this model yields a global hexagonal lattice that emerges from these pairwise interactions that are confined to 2D, whereas in 3D, the exact same model yields only local structure or local distances or local metric of space, if you will, but no global lattice, as in our bad data. And I just want to point out that a lack of a global lattice in 3D grid cells was found also in rats from the lab of Kate Jeffrey, also was just published. And the implications of these results, I think, are quite important for grid cells because this local metric of space in 3D argues against models of grid cells that rely on having precisely periodic firing, at least in 3D. So there are many models of grid cells, and the vast majority of them on the function of grid cells. And the vast majority of these models rely fundamentally on this precise periodicity. And if it's not precisely periodic, then the model just don't work. So really, this argues against many of the kind of geometric style models of grid cells. This also suggests that grid cells may underlie short-range 3D navigation because the short distance kind of metric property is preserved, but not long-distance navigation because the long-distance structure is not there in 3D. On the other hand, on the mechanistic side, I would say there is an interesting challenge for the field. There is strong accumulating evidence that grid cells emerge from continuous attractor dynamics. That's from experiments in rodents. But the types of attractor models typically show a kind of a rigid hexagonal type of lattice in 2D. So I think the challenge for the field is to develop models that combine the concept of a pairwise interactions, as we did in our model, and the concept of continuous attractor network. Can these two type of models live together? And at least the theoreticians, Chaim Tzompolinsky and Yoram Borak, think that the answer is yes, and they start working in this direction. So I think this will also mean that new models will emerge for grid cells. Okay, let me look at the time. Okay, sorry for that. Okay, the next story that I want to tell you about is the presentations of very large spaces in the brain. This is work by Tamir Eliav, together with Shir Maimon, PhD students in the lab. So bats navigate in the wild hundreds of meters underground, as you can see in this cave system, and also many kilometers outdoors, as I just showed you. And this serves sort of to illustrate the problem, because if you look at this flyway that I just showed you before, and this sort of defines a particular volume, right? You have, in this particular case, 15-kilometer long flyway. It's 2 kilometers wide and maybe half a kilometer high. This is roughly the range of altitudes that we've seen using the GPS tracking for the flights of bats outdoors. So this defines a particular volume. And if you want to now take your laboratory size place fields that you find in small laboratory boxes that are typically 10 or 20 centimeters in size, and use those 10 or 20 centimeter size fields to tile this volume of 15 kilometers by 2 kilometers by half a kilometer, then a little back of the envelope calculation will show you that you need about 10 to the 15 or so neurons. So depending on some assumptions. And now the entire number of neurons in dorsal hippocampus, area CA1, where we are recording here, is about 10 to the 5 neurons. So we are 10 orders of magnitude off. That means that this is not going to work. 10 orders of magnitude is a huge number. It means there's no way that what we find in small laboratory environments will translate one-to-one to what's going on outdoors. So to understand, then, I mean, this means there must be some other type of coding in for large spatial scales. And to understand this type of coding was the goal that Amir has set. So for this, we had to solve several technical problems. One is to be able to record wirelessly at essentially unlimited distances. So we developed these neural loggers, devices that record from the, we have a tetrode microdrives, we have tetrodes in the hippocampus in this case, but we store the data, the neural data on board the animal. And in this manner, we are essentially unlimited in distance. And here you see an example of nice spikes. These are the 4 channels of one tetrode, and you see really nice spikes that you can record in flight from the hippocampus of a flying bat. In the last couple of years, we moved to from a 16-channel neural logger to 64-channel neural logger into 16 tetrodes, which means now we can record populations of neurons. We can do decoding, all sorts of things that I won't go into, but that we are heading more in the direction of population recordings in flying animals, freely flying animals. We also have on board the neural logger an ultrasonic microphone, an 9-axis motion sensor, and we're adding more and more behavioral sensors. So that's basically an entire onboard flying neurophysiology lab, as opposed to the classical approach of experimentation where the animal is sort of running in an arena and everything, the cameras and everything is external. Here, everything is on the animal. The other thing is that we had to develop ways to measure the position of the animal very precisely, because if we want to dissociate between 10-centimeter place fields and other things, we need to be able to measure the position of the animal with 10-centimeter precision. So we found this solution from a French company that using an RF tag to measure distances to a local array of antennas. So we deploy these antennas, and we can measure now a distance to the bat. So this creates these circles, are essentially the measured distances, and where all these circles cut each other, that defines the position of the animal. And this indeed provided about a 10 or even 9-centimeter precision, which is much better than regular GPS. And the third is that we needed to build a large but controlled behavioral setup. So we built this type of a tunnel. You see it's 200-meter long. This is the end of the tunnel. And we put in there landmarks. We are, by the way, now finishing to extend this tunnel to almost 700 meters long. So really a very large behavioral setup. And in this tunnel, 200-meter long tunnel, the bats really flew beautifully back and forth. We put food rewards at both ends of the tunnel. And you see here time versus position, the tunnel of the bat, and the bat flew back and forth and back and forth and back and forth and back and forth, really flew many times. It was mostly driven by their will to fly. I mean, many of them didn't even eat that much, didn't care so much about the food. They really like to fly, and we suddenly provide them this enormous space to fly, so they do what they like, which is to fly a lot. Often 100 laps, 100 flights per recording session. So this could be 20 or even 20, almost 25 kilometers of flight in one recording session. So really very large distances. And they flew in a really one-dimensional fashion. So this is a cross cut through the tunnel. So the tunnel really looks like that, like a child's drawing of a house. This is really how the cross cut of the tunnel looks like. And you see that the bats really flew in the center of the tunnel and didn't deviate much in the z or the y dimensions. So really, we turned it into a one-dimensional problem, which was on purpose in this case. And they also flew very fast. In this case, the bat, this is data from one session. The bat flew at 7 or so meters per second and a very stable flight speed along the entire tunnel. Really, they maintained flight speed along the entire tunnel. We also quantified this, but this will be important in a second, that the flight speed was really extremely stable along the entire tunnel. And so in this case, we are utilizing the bat's flight speed to measure the representation of large spaces. Because the bat fly at 7 or 8 meters per second, we can have them repeat this 200-meter distance or 400-meter back and forth many times to yield reliable rasters. So you were utilizing really the bat's flight speed here to ask this type of question. And here's an example neuron that Tamir recorded. In the setup, what you see here is all of this is one neuron. On top are the PSTHs or firing rate maps of the neuron versus position. And at the bottom are the two rasters. So each line here in the raster is a different flight. And we've separated the two flight directions, the direction flight to the east in blue and flight to the west in red. And you see several things here. First of all, that the raster or the firing for the two directions is different. The red and blue raster are different from each other. And that's well known for many years, also in laboratory environments. Running in one direction versus running in another direction leads to a different map for the same neuron in the hippocampus. What's more interesting and new was several things. First of all is that the neurons could have very large place fields. So here you see in the blue raster that this neuron has maybe 20 or 25 meter size place fields, a huge place field. Not 10 centimeters. So there's definitely scaling up with the environment size. Secondly, there are many place fields. The neuron is active not in one location as we typically think about place cells, but in multiple locations. You see these multiple bands. And thirdly, that and maybe most surprisingly for us, is that the neuron had this multi-scale property. So the same neuron could have a large field in one location, like here, and a small field in another location. Here's another example neuron that has in the blue raster, it has a large field here, small fields here, and a medium field over here. So it has different sizes of fields in different locations. Okay, so this sort of summarizes these observations that the spatial code in this very large environment consisted of large fields, also small fields, but in particular, we had some very large fields, multiple fields for the same neuron, and this multi-scale property that the same neuron can have a very different resolution, if you will, in different locations in the tunnel. And this looks very different from place cells recorded in small lab setups, and we also ourselves recorded bats in the same tunnel, but where we blocked a 6-meter piece of the tunnel, and then we find the classical single place field thing as has been found for many years. So really, there's something different about large spatial scales. And this summarizes the population data that neurons have many fields. Some neurons had 20 or even 10 or even 20 fields per direction, so really many place fields. And this shows this multi-scale property, that if you look at the field size ratio for the same neuron, so what is the largest field divided by the smallest field of the same neuron, this is often 5 or 10 or even goes up to 20. 20 means that the same neuron could have a 1-meter size place field here and 20-meter size place field there. So really a very surprising result. We had some controls that I won't go into. We could show that fields do not cluster near landmarks. And in particular, small fields do not cluster near landmarks. So it's this multi-scale property is not driven by the landmarks. Flight speed was stable along the tunnels. It's not that there was a variation in flight speed that could explain this different scales, but in fact, really the flight speed was stable. Also, it could not be explained by spike sorting or definitions of fields. We had controls for all of that. So really, this multi-scale coding is a genuine phenomenon. Shir Maimon, another PG student in the lab, then asked, are these multi-scale, does this multi-scale code require prior experience? So she did two types of experiments. One, she actually recorded from day one in the tunnel. So from the very first minute the bats were exposed to this tunnel, and found that the multi-scale code is seen already from day one. So it does not require prior experience with this particular tunnel. And moreover, and perhaps more surprisingly, she looked at lab-born versus wild-born bats. So the data that I showed you so far, and actually all the data I'm showing you today, except this, is from wild-born bats. So bats that were born outdoors, grew up, and we caught them as adults in the wild. So they've definitely seen and flew over kilometer scales. But we also have a colony in the lab, and we have bats born in the lab, and they actually fly quite a lot. So they're in good shape. It's not that they're sort of couch potatoes, they don't fly. They fly very well, but they've only fly kind of locally in a small room. It's about 7 meters or 6 meters. So these lab-born bats, we expected actually to show a different type of coding, but surprisingly enough, the codes seem to be very similar. So they're looking at the number of fields per direction for the wild-born versus lab-born bats was very similar. Also, this field size ratio between the largest and smallest field was similar. So it looks like there actually wasn't a difference between the lab-born bats and the wild-born bats. So that means that this type of multi-field, multi-scale coding that we observed is very robust. It's there regardless of experience. It seems to be a very basic property of the circuit. And again, we asked, why is this? It's a very surprising type of coding with sort of multiple receptive fields, each of a different size. Think of something like that. And for a V1 neuron, this would sound very strange. So why is something like that useful? We again teamed up with the theoreticians, Yonatan Al-Khadef and Misha Tzodiks. And again, we did a lot of things. I won't go into all the details, but I'll just give you a flavor of the type of approach that we took. We took a decoding approach where we compared various coding schemes. So essentially, scheme 1 is the classical place code, where we have, in this case, 10 neurons. These 10 rows are 10 neurons, and these are 200 meters. So each neuron has a small place field in a different location. So this is the classical place code with small fields. Then we can look at neurons with large fields, or neurons where field size varies, for example, with anatomy, and that's actually, we know that to be the case in rodents in small environments. And then all the way to multi-field type of codes, as in this scheme 5 and 6. So the difference is that scheme 5, each neuron has multiple fields, but all of these fields are the same size for the same neuron, unlike what we see in the bats. And scheme 6 is basically what we see in the bats. So each neuron has multiple fields, and they each have different sizes. And we did decoding analysis of the position of the animal in the tunnel in simulated bats using these various encoding schemes. And what we found is that looking at probability of catastrophic errors, where we define catastrophic errors as a decoding error larger than 5% of the environment size, versus various environment sizes. We simulated from 20 meters to 1,000 meters environments, right? In simulation, we can test all possible sizes. What we found is that in small environments, there was no difference between the various coding schemes, but in large environments, there was a big difference. In fact, the multi-scale coding scheme, which is what we found in the bat, was almost 3 orders of magnitude better than the classical place code here in red. So really, it seems to be that this multi-field, multi-scale code is much, much better for large environment, but not necessarily for small environments. And this very low probability of decoding errors was achieved with just 50 neurons. So really a very small number of neurons. You can represent very well huge environments of up to almost a kilometer. So to summarize this part, spatial coding of large spaces is very different from lab setups. We have many fields per neuron, often more than 10 fields in each direction. We have very large fields in dorsal C1 neurons up to 30 meters, but also very small fields down to 1 meter or even less. And a given neuron exhibits multi-scale spatial coding where the same neuron can have field sizes that differ by up to 20-fold from each other. And this multi-field, multi-scale coding is independent of experience, and theoretical analysis of decoding errors explains why multi-scale coding is better for large environments. Okay. Let me switch gears. I'll run very quickly through this part and then tell you about the last part. So this third story I'll tell you very, very quickly about is social representations, work by David Omer, a former postdoc in the lab. Now he has his own lab in the Hebrew University. So basically the question that David asked is, because bats are very social animals, can you find in their hippocampus where there are place cells, neurons that represent my position, are there also neurons that represent your position, the position of another individual in a social setting? So you devise this experiment, kind of a mimicry task with two bats, observer and a demonstrator. The demonstrator bat was flying from one ball to another ball, and the task of the observer was to mimic it. So if the blue bat flew to ball A, then the red bat is also supposed to fly to ball A, and if it goes to ball B, it's also supposed to fly to ball B. So really, they're flying in an alternating manner, but we're mimicking each other. The red bat was mimicking the blue bat. And just to show you one example neuron, on the left here is the self-flight, so kind of the classical place representation where when I'm flying, the observer was flying, you see that this neuron fired on the left arm in a particular location, forming a nice place field. But when the other bat was flying, the observer was stationary, and in fact, we had an accelerometer to verify it really does not move at all. Still, the neuron represented the position of the other bat, and had a nice place field. In this case, the place field for the other bat was different than the self place field. So we call these neurons that represent the position of another bat when I'm stationary, we call them social place cells, and we can rule out trajectory planning type of explanation. We also asked, is it really representing the other conspecific as opposed to objects? And we did experiments with objects as well, and we were able to show that really the responses to objects are very different. So we think that these type of representations of the position of another individual may be important for social spatial cognition, like representing conspecific, predators, prey, etc. We as humans are obviously very good in representing and following the positions of others in a party or in the park with our kids. We know, where they are, or of course in ball games like football or basketball, the players are really good at knowing where are other team members and opponents. So perhaps these type of neurons can underlie these behaviors. And just to tell you that we are now running a really crazy experiment, but a very fun and promising one, I think, by a postdoc Saikat Ray, where we created a bat colony where we have between 5 and 10 bats living together 24/7, creating a social network and interacting between each other. And here we can ask, we found ways to track the positions of identities of all these 10 animals simultaneously in the room, and record neurons from these flying bats. You can, of course, when you have wireless methods, you can easily record. You don't get cables entangled with each other. And then we are asking, is there a representation of the identity of another individual, a representation of males versus females, representations of my friends, the bats I like to interact with most, representation of particular types of social interactions. And we sort of see also very interesting things. This is ongoing work, and I hope that within a year or so, we will have a clear story, but we already see very interesting neuronal responses in this very naturalistic type of situation. And the last story that I want to tell you in really a few minutes is about natural switches in behaviors that rapidly modulate position by distance coding by hippocampal neurons. This is work by Ayelet Sarel and Shaked Palgi and Dan Bloom. So sort of the rationale for this for this question is that, if when I go from home to work, for example, then initially I navigate. It's just a few minutes walk. So I navigate. Then I get to a T-junction. I need to decide whether I go right or left. Is the decision making involved? Then I might bump into somebody and talk to them, to the social interaction. So within just a few minutes of going from home to work, I might switch from navigation to decision making to social interaction. I keep switching between things. And so the overall problem is that we, in real life behaviors, we dynamically and rapidly switch between different behaviors, but that's not how we do neuroscience research. Neuroscience experiments are typically static in the sense that we study one behavior at a time. It'll be a social neuroscience experiment or a navigation experiment, just different types of experiments. So how does the brain deal with this behavioral dynamism? That was the general question, and specifically in the context of navigation, when we navigate in transit, encounter something like a conspecific and direct our attention to it, do hippocampal neurons rapidly switch or present this new thing out there, and how does it work? So this was the general question, and so to address this, we did this two-bat experiment in the tunnel, where we had bats either fly individually in solo flights or fly opposite each other. And these flights, these solo flights and these crossover flights were intermingled. So these were not two different sessions. It was all one session, but sometimes one bat was hanging and the other bat was flying solo. Sometimes they were flying opposite each other. So there was this intermingled behaviors. And the positions, the encounters of these crossovers here happen in all possible positions along the tunnel. So we get a nice coverage of the two-dimensional plane of allocentric position by egocentric distance from the other bat. And you see it here. This is the position of the bat, and this is the distance between the bats. And we have a nice coverage of this plane. And this is already responses of two neurons. This particular neuron got strongly activated in a particular position in the tunnel, but also in a particular distance from the other bat. So there's sort of a conjunction of position and distance. This neuron actually shut down at a particular distance, was inhibited in a particular distance. So we see, and we had controls that these neuronal responses could not be explained by changes in speed or in lateral deviation. And this shows the population from 4 bats. This is kind of the tuning curve. Each line here is a tuning curve of one neuron. And you see that there was a sort of an overrepresentation between -20 to +20 meters. And the interbat distance at which neurons sort of started to represent the distance to the other bat was about 15 or 20 meters, which was the distance at which the bats switch their attention as indexed by the increase in sonar rate or echolocation rate. And what do I mean by that? If we look at the echolocation behavior of the bats, this now is a raster of echolocation behavior, not neural raster. Here we have 52 trials, meaning 52 crossovers in one session. This is the interbat distance. And the dots here are the echolocation calls of the bat. And you see that at a distance of 15 or 20 meters, they increase the rate of echolocation calls, which we know for many years is an index of the bat's attention. You see this increase in rate. And so it's nice. We have a microphone in the bat's head, and we have an independent behavioral index of attention to the conspecific which we can measure on a trial-by-trial basis. And indeed, Shaked Palgi in the lab found that by separating flights with high click rate or high attention versus flight with low click rate or low attention, neurons were modulated by this separation. So the firing rates of neurons was oftentimes higher for the high click rate flights versus the low click rate flights. You see here 2 example neurons, and this is the population showing a systematic difference there. So really, attention modulates the activity of the neurons on a trial-by-trial basis. This cannot be explained by simple auditory responses. Again, I won't go into the controls. We also found that different place fields show different distance coding. So here is an example of one neuron here on the left. So you see that in this field, in this place field, again, this is position versus distance. In this place field, it got activated. You see it in the raw data and in these purple spikes. It got excited. In the middle place field, it actually didn't care. It was flat. in the lower place field, it also got excited. So the same neuron could either respond or not respond in this case, depending on which place field you're in. This is another neuron that in one place field got excited, in another place field it got inhibited. So totally opposite behavior of the same neuron. And we looked at the correlation between these in-field tuning curves, distance tuning curves, between pairs of fields in purple versus shuffles in gray, and these distributions are quite similar. So it looks like the distance tuning within different places of the same neuron are almost independent. That means there is a non-separable representation of position by distance. So the distance coding in different positions in tunnel is different. And theoretical decoding analysis by Yonatan Al-Khadef showed that such a non-separable representation improves coding. Again, don't have time to go into this. So to summarize this story, during crossovers, many place cells in hippocampal area C1 transiently encoded the distance from the other bat. This neuronal representation of the other bat was very brief, lasting for only a few hundred milliseconds, right? Because the bats are flying. This one is flying at 70 meters per second. This one at 70 meters per second. So it's 14 meters per second combined speed. It's really fast. And as they approach each other, there's this switch to representing the distance to the other bat, and then they pass each other and it goes back to baseline. This all happens very fast. So the switch from allocentric encoding to allocentric by egocentric encoding. And these results suggest that neural circuits can modulate very rapidly the core computation that they perform. That is, that neural coding can be very dynamic. And we also found modular computations in different place fields. So different place fields show different types of distance coding. So to summarize overall what I showed you today, we discovered various types of novel types of coding when incorporating natural aspects of behavior, right? We looked at movement in 3D, and then we found that grid cells look completely different, have this local coding, but not the global lattice. Also, the head direction cells were very interesting with this toroidal representation that I mentioned. When we look at natural spatial scales, and everything looked completely different, multiple fields, multiple scales for the same neuron, totally different type of coding. Social interactions, we found interesting representations. Also, natural switches between different behaviors. We see great dynamism and what the individual neuron can do. So really, my sort of last slide and the overall take-off message is about the importance of natural behaviors. So, we have this tradition of reductionism in biology and in science in general. And of course, reductionism has great strength. But I would argue, I always worry that if we reduce too much and control too much, then we might miss the very thing we're trying to investigate, or to understand, which is behavior, because behavior is rich, is complex, is context dependent. And if we simplify things too much, we may miss what we're looking for. And so, of course, on the other hand, reductionism has its powers. So I'd like to argue that we need diversity, both the classical reductionist approaches, but also more naturalistic behaviors. And as I showed you today, every time we go into some naturalistic behavior, naturalistic scale, social interactions, dynamic behavior, etc., we find something interesting about the brain. And I'd like to argue that this can be extended to all of neuroscience. Basically, I'm now also writing a book for MIT Press called Natural Neuroscience, where the argument is exactly this, that in every domain of neuroscience, you should go for more naturalistic behaviors. So I told you today a little bit about neurobiology of navigation, what you can find when you employ more naturalistic behaviors. But this is true for active sensing, sensory neuroscience using natural stimuli, real-life memory research, decision-making, social neuroscience, et cetera, et cetera. And with this, I'd like to finish and thank the lab members, and in particular, Lior Alas. She's a fantastic staff scientist working with me. She's been involved in everything that I showed you today, so I really want to thank her. And also the lab members that I think I mentioned, most of them as we went along, and alumni and funding sources. And thank you for listening.